Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's lesson, this will be the part 2 of organic compounds which are essential to human life. And I'll be starting this lesson with proteins' function as enzymes. So stay tuned. In this picture are the steps in an enzymatic reaction. In figure A, substrates approach active sites on enzyme, and in figure B, substrates bind to active sites, producing an enzyme-substrate complex. And in figure C, changes internal to the enzyme-substrate complex facilitate interaction of the substrates. And in figure D, products are released and the enzyme returns to its original form, ready to facilitate another enzymatic reaction. So I'm gonna start this lesson with proteins which will function as enzymes. And if you were trying to type a paper and every time you hit a key on your laptop, there was a delay of 6 or 7 minutes before you got a response, you would probably get a new laptop. And in a similar way, without enzymes to catalyze chemical reactions, the human body would be non-functional. It functions only because enzymes function. So an enzymatic reaction is a chemical reaction catalyzed by enzymes, and it begins with a substrate bind to the enzyme. So what is a substrate? A substrate is a reactant in an enzymatic reaction. This occurs on regions of the enzyme known as active sites. So any given enzyme catalyzes just one type of chemical reaction. This characteristic called specificity is due to the fact that a substrate with a particular shape and electrical charge can bind only to an active site corresponding to the substrate. So binding of a substrate produces an enzyme-substrate complex. It is likely that enzymes speed up chemical reactions in part because the enzyme-substrate complex undergoes a set of temporary and reversible changes that cause the substrate to be oriented toward each other in an optimal position to facilitate their interaction. This promotes increased reaction speed. The enzyme then releases the product and resumes its original shape. The enzyme is then free to engage in the process again and will do as long as substrate remains. And here are the other functions of proteins. Advertisements for protein bars, powders, and shakes all say that protein is important in building, repairing, and maintaining muscle tissue. But the truth is that proteins contribute to all body tissues from the skin to the brain cells. Also, certain proteins act as hormones, chemical messengers that help regulate body function. For example, growth hormone is important for skeletal growth, among other roles. As it was discussed in the previous video, the basic and acidic components enable protein to function as buffers in maintaining acid-base balance, but they also help regulate fluid-electrolyte balance. So proteins attract fluid, and a healthy concentration of proteins in the blood, the cells, and the spaces between cells helps ensure a balance of fluids in these various compartments. And moreover, proteins in the cell membrane help to transport electrolytes in and out of the cell, keeping these ions in a healthy balance. So, like lipids, proteins can bind with carbohydrates, and they can also thereby produce glycoproteins or proteoglycans, and they both of which have many functions in the body. Like the body can use proteins for energy when carbohydrate and fat intake is inadequate, and stores of glycogen and adipose tissue become depleted. However, there is no storage site for protein except functional tissues. Using protein for energy causes tissue breakdown and results in body wasting. The fourth type of organic compound important to human structure and function are the nucleotides. So what is a nucleotide? A nucleotide is one of a class of organic compounds composed of 3 subunits, and these 3 subunits are one or more phosphate groups, that's number 1. Number 2 is pentose sugar, either deoxyribose or ribose, and the third one is the nitrogen containing base adenine, cytosine, guanine, thymine, or uracil. Nucleotides can be assembled into nucleic acids, DNA or RNA, or the energy compound adenosine triphosphate. In this picture are the nucleotides. In figure A, the building blocks of nucleotides are one or more phosphate groups, a pentose sugar, and a nitrogen-containing base. And in figure B, the nitrogen-containing bases of nucleotides, and in figure C, the 2 pentose sugars of DNA and RNA. Let's move on to another topic, which is the nucleic acids. The nucleic acids differ in their type of pentose sugar. Deoxyribonucleic acid, or DNA, is a nucleotide that stores genetic information. DNA also contains deoxyribose, so-called because it has one less atom of oxygen than ribose, plus one phosphate group and one nitrogen-containing base. The choices of base for DNA are adenine, cytosine, guanine, and thymine. Ribonucleic acid, or RNA, is a ribose-containing nucleotide that helps manifest a genetic code as protein. So RNA contains ribose, one phosphate group, and one nitrogen-containing base. But the choices of base for RNA are adenine, cytosine, guanine, and uracil. The nitrogen-containing bases adenine and guanine are classified as purines. So, what is a purine? A purine is a nitrogen-containing molecule with a double ring structure which accommodates several nitrogen atoms. So, the base is cytosine, thymine, found in DNA only, and uracil, found in RNA only, are pyrimidines. So what is a pyrimidines? A pyrimidine is a nitrogen-containing base with a single ring structure. Bonds formed by dehydration synthesis between the pentose sugar of one nucleic acid monomer and the phosphate group of another form, which is a backbone from which the component's nitrogen-containing bases protrude. In DNA, 2 such backbones attach at their protruding bases via hydrogen bonds. This twists to form a shape known as a double helix. The sequence of nitrogen-containing bases within a strand of DNA form a genes that act as molecular code, instructing cells in the assembly of amino acids into proteins. Humans have almost 22,000 genes in their DNA, locked up with the 46 chromosomes inside the nucleus of each cell, except red blood cell, which lose their nuclei during the development. And these genes carry the genetic code to build one's body and are unique for each individual except identical twins. So in contrast, RNA consists of a single strand of sugar phosphate backbone studded with bases. Messenger RNA is created during protein synthesis to carry the genetic instructions from the DNA to the cell's protein manufacturing plants in the cytoplasm, which is the ribosomes. In this picture is the structure of adenosine triphosphate. The nucleotide adenosine triphosphate, or ATP, is composed of a ribose sugar, an adenine base, and 3 phosphate groups. ATP is classified also as a high-energy compound because the 2 covalent bonds linking its 3 phosphates store a significant amount of potential energy. And in the body, the energy released from these high energy bonds help fuel the body or the body's activities, from muscle contraction to the transport of substances in and out of cells to anabolic chemical reactions. So when a phosphate group is cleaved from ATP, the products are adenosine diphosphate or ADP and inorganic phosphate. Removal of a second phosphate leaves adenosine monophosphate or AMP and two phosphate groups. Again, these reactions also liberate the energy that had been stored in the phosphate bonds. They are reversible too, as when ADP undergoes phosphorylation. So what is a phosphorylation? A phosphorylation is the addition of a phosphate group to an organic compound, in this case resulting in ATP. In such cases, the same level of energy that had been released during hydrolysis must be reinvested to power dehydration synthesis. Cells can also transfer a phosphate group from ATP to another organic compound. For example, when glucose first enters a cell, a phosphate group is transferred from ATP, forming glucose phosphate and ADP. Once glucose is phosphorylated in this way, it can be stored as glycogen or metabolized for immediate energy. So this ends my lecture about the part 2 of organic compounds, which are essential to human life. Once again, if you have any question, please write it in the comment section down below and I'll be glad to answer them all. And thank you so much for watching and for listening to my discussion. Until my next video, bye everyone!